Alright, so the next thing that we'll talk about is citric acid cycle, but we need to do a little bit of a prep step first that we call pyruvate oxidation. And remember, oxidation is a loss in electrons, so we'll be stripping some electrons away from pyruvate during this process. Pyruvate is that 3-carbon compound that we got at the end of glycolysis. It will be transported into the mitochondrial matrix, and so we'll have that outer membrane of the mitochondria as well as that highly folded inner membrane, those cristae. So we're all the way in the middle here in the matrix region. So within the matrix, what we're going to do first is go ahead and strip off some electrons from pyruvate. We have our C3 molecule moving in, and we're going to split off and strip electrons from that and give those to our electron carrier NAD+ to reduce it to NADH. Recall that for each glucose that originally came in, this whole process is going to happen twice. So, for each of the pyruvates that were made, this will happen. Once we get the NADH and those electrons stripped off, a carbon dioxide is going to be taken out, and we're going to join to a coenzyme. Coenzymes do not get used during the reaction, but they do kind of help the enzyme in different ways. So this coenzyme is coenzyme A that will come in. And so that 2-carbon molecule that we have plus that coenzyme is referred to as acetyl for the 2-carbon, CoA for that coenzyme. So this is what goes into the citric acid cycle, is this 2-carbon compound. The 2-carbon is going to join up with a 4-carbon, which is oxyl acetate, and it joins with this 2-carbon. So 2 plus 4 gives us a 6-carbon, and this is what gives the cycle its name, the citric acid cycle. This 6-carbon molecule is citrate. Then what we're going to do with the citrate is we're going to rearrange it a little bit. We're going to actually produce some water, but that's going to go right back in. So we don't really consider that a product here. So the citrate rearranges to another C6 molecule. It rearranges to another C6 molecule. And then this is finally where we start stripping out some energy. This C6 molecule here, we're going to go ahead and strip off some electrons and give it to our electron carrier. Okay. So we're going to couple that with a breaking off of carbon dioxide. And carbon dioxide, because it's a small nonpolar gas, is going to go ahead and diffuse right out of the mitochondrion and out of that cell. We're going to go ahead and strip electrons at the same time and give those to our electron carrier NADH. So we go from here, a C6 molecule to a C5 molecule. We're going to do that same process again and strip off a carbon dioxide. That will give us a C4. And then that C4, we're going to couple that with providing electrons to our electron carrier NAD+ to NADH. We're going to do some rearranging, and during that rearranging, we're actually going to make some ATP, and that will be due to what's called substrate level phosphorylation, which basically means that we're going to use an enzyme to do that. We saw that occur in glycolysis as well. All right, so we have a C4. We're going to continue to manipulate it to get some more electrons out. All right, I had to do a little bit of shuffling there, but now we're on to C4. We're just rearranging. And during the process here, we're actually going to get out an electron carrier that we haven't seen before. FAD is going to be reduced to FADH2. Another rearranging. And here we're going to get out our final electron carrier, NAD+, to be reduced to NADH. And we've completed our cycle. We have our C4 back, that oxyl acetate that we started out with. So during this process, we've made 1, 2, 3 NADHs. We've made 1 ATP, and we've made 1 FADH2 from a single pyruvate. So you would just double all of those 4 products at the end of the citric acid cycle. Now, the next step, we're going to do work with all of those electron carriers that we have just made.